Right. Let's continue our discussion about the normalization in single-cell RNA-seq data analysis. As a follow-up from our last lecture, right now we are at the second step for the whole pipeline. So we talked about the quality control last Wednesday, and then on the past Monday, we started talking about the normalizing data step. So the default, just as a quick recap, is, let me try to pull up the laser. The recap is, the default is the size factor normalization, or you may just call it log 1p CP10K. So a recap is that we normalize each cell total read count, total UMI count from whatever number it used to be to 10K as the new total. And then with the scale new total, we add 1 to it before take the log. So we will provide some insights about why this is reasonable today. But this is what's in SURAT normalized function, and in ScanPy's normalized total and log1p function. All right. Then last time we said that in this bioRxiv paper, it compares this log normalization approach, the default one, with the SC-transform approach, which is the right one. So we will talk later about this. So because there's still some discussion about which one is better. So the SC-transform was proposed later than the default. And it argues that SC-transform can remove the size pattern within a cluster. So you don't see a gradient that follows the size pattern, the original size from the smallest to the large, from the small to the large on this side. But you will see that in this monocyte cluster, you still see some pattern. So yeah, we'll talk about this bioRxiv preprint version. So what is SC-transform? I think we stopped here last time. It has multiple steps. The first step is that it tried to fit a regression model called the negative binomial generalized linear model. So in the notation, which we discussed last time, Xi stands for the unnormalized, the original UMI count of gene I. And we assume that the gene I follows a negative binomial distribution for the mean, which is actually denoted by mu down here, but the connection has to be inferred by the readers because the manuscript didn't, the publication didn't say EXI is equal to mu or mu I for gene I, but it is. So basically, we're just transforming this negative binomial mean by log, assuming that the log mean is a linear function of there's only one feature, one covariate, one variable, or one predictor. So people use different words in different settings, but in general, in machine learning, you may call this a feature, but when you do regression, the more common ways to call this covariate. So this covariate is the total UMI count of one cell the gene is in. If one cell taken log transformation, log 10 transformation. So you see the reason why SC-transform wants to do this regression is try to remove the effect of the cell library size or the total UMI count effect. Because basically you want to say that this is the relationship so that for each gene I, its mean depends on this total library size in the cell. And as the cell gets a larger library size, I expect a bigger mean. But this is not what I want, right? You see, in this default approach, what you effectively, what you change or what you remove is the original total library size, which is this, the summation of gene J prime from 1 to P for the gene I in, oh, sorry, my bad. The summation of gene J high prime from 1 to P for cell I. So you're taking this total for cell I, but you're changing it to 10K. So you remove the original cell size and you do the same thing in SC-transform, but through a regression framework. So the goal is that the model can explain the general negative binomial generalized linear model can explain the effect of cell library size on gene I's count. So you know there's some swapping between the I and J. So in the assay transform, I is for gene, but previously I is for cell and J is for gene. So you have to be very careful about which one the index refers to. Then the hope is that once we fit the model, you can assume that we have a line like this. Let me see if I can draw something. So for any regression framework, you would have this log 10. I'll just add back the notation for cell J. So this is cell J's total library size log transformation. And what you have is this log, basically, expectation is the mean, negative binomial mean of gene I in cell J, Xij. And you're going to fit a line for each gene I. So maybe the line is something like this, a linear line. So on the log scale, this is what you have. Then what you can see is that, okay, if I get rid of the log on the Y scale, I can have a relationship between just expectation of Xij and log 10 MJ. So for that, you might have something like an exponential curve, right? Because right now it's a linear curve. After you take the log out of Y, you get an exponential term. Then you will just compare your Xij, your observed Xij to this line, and you take the residual. And the residual means that it's the remaining part of your original Xij after we remove the size fact, the library size effect. That's the whole rationale. Okay, so basically, let's look at the specifics in the paper. So the procedure we developed has 3 steps. In the first step, we fit independent regression models per gene. So that's what I said. So you will have this model 1 per gene I. So that's why last time I said your coefficients beta 0, beta 1 should become beta 0I, beta 1I, because they are specific to gene I. So we should add it here. Okay. And in this line, the index of cell J was not included, but it was in the text, okay? And then when you say the negative binomial is for gene I, then this mu should be mu I. And then sig and the theta for dispersion, they should be theta I. So they should all be like this. So in other words, what we are assuming here is that Xij, yeah, it's not easy to use both the it's not easy to use both the PowerPoint and writing, so I'm just trying. So basically, you assume that for Xij, they are following across all the J's. For each J, they are independently following a negative binomial distribution. So I'm not writing IID because it's not identical distribution, but they are independent distribution with mean mu ij, mu I. So it should be mu I. And the negative binomials dispersion parameter is theta I. So this will together give me the variance, which we can written as theta I squared. Okay, so that's the negative binomial mean and variance. And specifically, for this mu I, will be specified by this log link function. So this is actually exponential function to the power of beta 0I plus this beta 1I times the log m. Yeah, so log mj, and j will enter here. So basically, this is the effect of the cell library size, and this is the linear part, and this is the exponential function to reverse the log, and that gave me the mu I. So basically, this is the definition of mu I. So even though we didn't actually talk about generalized linear models we haven't talked about this specifically in this class, I hope to give you a high level idea. At least for those of you who know what is generalized linear model, you can see the connection here. But based on what I wrote here, you could see how much information was missing in the original description. So the reason I could guess what it means, or I could understand this just because I understand generalized linear models. I could piece the notation together. But maybe you cannot, right? So that's why last time I asked you how many of you understand it, but I didn't get a hand last time. So that's why there are a lot of missing pieces in the notation, including I and J, including what is actually the model. Yeah, the model was not written down. And another reason, I think I had a discussion with a friend who we both feel like the notations are becoming more and more not detailedly written or sloppy. We became more sloppy nowadays. And one possible reason was because of people's reliance on software. So if you're using other people's packages, you may just implement it. But if you are doing methodology yourself, you have to derive from the scratch. You need to write down the notations correctly. Otherwise, you will confuse yourself, right? What I'm doing here. All right, so later we'll see another paper today, SCnorm, another normalization method, and we can compare the notations. All right, so coming here. So the reason I will give you this is because, all right, so actually there is mu xij, mu ij, theta ij. Yeah, I should say, my bad. Yeah, even I missed the thing here. It should be mu ij, sigma ij. Because why do I say mu ij? The definition here involves J, the J of cell J. So it will enter the mean. So what I mean here is that essentially we want to add J here and J here to mean this is the equation for cell J. Then this becomes mu ij. Okay, so this is the mean expectation for expectation of xij. Then you see, once you have these, then you just standardize your xij by subtracting the mean and divide it by its standard deviation. But another thing I want to make a note here is that when we talk about statistical model, these parameters are unknown. We don't observe them. And what we only observe is our data, right? The xij is what we observe. And from our data, we could never get the true values of the parameters. Instead, we could only get the estimates of the parameters. So because of that, to be exact, we should add hats to everything because you're not getting the true value. You're getting the estimate. So this should become hat, hat, and hat. Yeah, so everything needs to add a hat to it to make sure, okay, it's the estimate, it's not the parameter. In particular, you see here, the authors added back the INJ. When we talk, then they talk about the mu ij, right? So I would say when you fit a negative binomial generalized linear model, what you get out of it for each gene I will be the intercept beta 0 I hat, the estimate for the intercept, and then estimate for the slope, beta 1 I hat. You will get these two. And this cell J's library size is what you have in your data. So then you can compute mu hat ij in this way. Okay, so you get this numerator, xij minus mu ij hat. How about this? How about the sigma, this standard deviation, sigma ij for for the xij? So for that, you will also get it from the negative binomial generalized linear models. This is this term you will get out of it. So coming back here, suppose that when you fit the first step, what you will get out of it will be, like I said, you will get mu ij, this, and you will get sigma ij squared. So you will get both from the first step. But second step is what they called to avoid overfitting. We briefly discussed this last time. So what it actually means is that you may want to borrow information across your genes so your estimates, parameter estimates are more stable instead of varying freely. And this is the idea including DSEQ2 as well. I will show that very soon. So basically, when we just do the first step, we only use each gene I's own data. We didn't use other genes' data. But in the second step, when they say we explore the relationship model parameters model parameter values and gene mean to learn global trends in the data. We capture these trends using a kernel regression. This is the R function. We use a normal kernel, first select kernel bandwidth. This is the technical detail about how to fit it. But essentially, the second step is like this. And this is from the DSeq2 paper. So what they mean here is that for the dispersion parameter of the negative binomial, which is this theta, or which we also have here theta I. So you can write the negative binomial using two parameters, right? One is you can write it in another way based on mean and dispersion. So basically, this will give you Uij, mu ij, and this will give you theta I. So when we fit a negative binomial regression, we will get a mean of xij, which depends on j. So for each cell j, the gene I will get a different mean, but theta I is the common dispersion parameter shared by all cells for gene I. So you will have this theta I. All right. And then what they try to say here is they try to fit this thing. Okay, so this dispersion parameter estimate, if we still use the notation we get from before. So this is theta I hat for each gene I. And this is what? Mean of normalized counts. So probably we just say that this is just the mu ij hat. Maybe there's some other small changes, but the idea is that you will use the mu ij hat. And maybe you take the average across all the j of the cell. You take the average of that. Then you can get a dot for each gene. So every dot here is for one gene. So if you have, say, P genes, you have P dots here. So what you're doing here is that you additionally, in the second step, fit a line, a smooth line through between the dispersion and the mean. And the hope is that you will try to reduce your estimation for, reduce the variance of your dispersion estimates. So previously, they are basically, these black dots are your estimates from step one. But once you fit this line, you could have multiple ways to do it. For example, in R, you could use what the authors use, the k-smooth, or you could use the low S function. Low S is another thing. I think probably the DSeq2 use the low S. So this function will basically fit this line. It's not a linear line, but it's a locally smooth line. And through this, and then what it does is that you try to map every theta I hat to the line. So basically, vertically, you draw the dots to the line, and you will use the values on the red line as your new theta I hats. So you update your parameter estimates. That's what's done here. All right, and the idea here is that you want to basically say, okay, for all the genes, they share some common pattern between dispersion and the mean. And that's what I want to learn from it. And I skip some technical details because basically the language here, MLE, prior mean, MAP are all Bayesian languages, Bayesian statistical language. You can see here, MLE, maximum likelihood to estimate, MAP, maximum A posterior, and prior mean is what is specified by the red line. So it's a very, I would say, unconventional procedure, honestly, because when you do Bayesian statistics, you want to have your prior specified before you have the data. But here, you are actually learning the prior after your first step. The red line is considered to be your prior. And MAP, in the Bayesian sense, it can be regarded as a weighted average between your prior and your MLE. So MLE is purely from the data, and prior is from your prior assumption. And when you aggregate the information, you get MAP. So that's why essentially in DSeq2, you will use the values that basically every black dots will be drawn to the red line by different degrees. And where they end will be what you call the MAP, and you use those. Okay, so Something else the authors of DESeq2 play with is that they also exclude outliers. So basically, these dots were excluded from fitting this line. So there are many things involved in here. And I think back to SC transform, they didn't specify it very clearly. How did they exploit the relationship? But the basic idea is that once you get all the genes estimates, you try to do here. So the negative binomial dispersions are no longer based on gene I alone, but using other genes information. Okay, so let's come back here. So let's say once after step two for each xij, you will have some updated estimates for mu ij and sigma ij. And that's what you'll put into the last step, the normalization step. So they will try to define this as a Pearson residual because this formula was first proposed by Pearson 100 years ago, and the Pearson correlation was named after him. He was one of the founders of statistics. So essentially what it means is that you take your observed xij, you subtract the mean predicted from the MBA-GLM model, you divide by the standard deviation estimate from the negative binomial. You call this Zij. And this z-score is commonly referred to this formula. Whenever you see z-score, you mean subtracting mean, dividing standard deviation. Okay, and this is the output. Okay, this is the output of SC transform. This Zij. So we are no longer working with xij, but Zij. All right, yeah, so I'll skip those details for your own reading, but that's the basic idea. And the authors said that their approach was inspired by DESeq2, basically, because that relationship was used in DESeq2. And in this paragraph, negative binomial was from DESeq, and then DESeq2 extended to the negative binomial regression because DESeq, the first version, was not based on the regression framework. It's just negative binomial, then hypothesis testing. But if you're interested, I talked about it in my previous lectures, previous year. So there was a lecture talking about DESeq specifically, and why I think the hypothesis test within has some issues. I think maybe the authors also realized the issues. That's why DESeq2 used a totally different framework. The only thing that remains is the negative binomial assumption. They changed the DE problem to a regression problem instead of a hypothesis testing problem. So for hypothesis testing, you need to come up with a test statistic derived as null distribution under the null hypothesis. That's what's in DESeq. But I think there's some issues with the null distribution or but how does the test statistic how is the test statistic distributed under null? Because it was not a standard test. It was not like a t-test or t-test, Wilcoxon test, not like those. It was totally derived by the author. So I have some questions about that. But then DESeq2 changed to a more standardized approach, the negative binomial GLM. Yeah, but this shrinkage step is specific in DESeq2. So the authors added this. Maybe they realized that, okay, for RNA-seq DE problem, typically there are only 3 samples per condition. So when you fit negative binomial regression model for 3 plus 3 data points, your parameter estimates are not stable. I think that was the major reason why they want to propose this shrinkage thing. And by shrinkage, I should say, empirical-based shrinkage, this is a statistical jargon. What it actually means is this. You try to draw your parameter estimates to some predefined pattern by shrinking them to the pattern. So they are less variable. You are shrinking the variances, right? Because you have a bigger scatter of your Ys along the line, but now after the shrinkage, they're more closely crowded, right? They're basically close to the line. That's what I mean by shrinkage. So it's a technique to stabilize the estimates by borrowing information from other genes. Okay, and HR also uses negative binomial GLM, but they may have some differences in the implementation. Okay, so basically you should assume that methodologically, HR and DESeq2 are similar. However, in practice, you may see that they sometimes give you very different results. Yeah, so that's the thing we have to be careful about. So I don't know what you think after this. Do you have any questions before? Yes. Sorry, could you speak loud? Why the author would use what function? Totally. Exactly. I think it's a very, very good question. I assume that you must have some background about Bayesian inference. The question to people on Zoom, the authors use a low S function in this way instead of specifying a full Bayesian model, right? From the beginning, you just specify the dispersion and the mean has a relationship, and then you estimate that model. Very good question. Actually, this was a question proposed to me. You know, UCLA every summer has the CGSI, Computational Genomics Summer Institute. So I met a faculty member there. He's from Israel, and Sahara Wasset. He's a statistician from Israel. He mentioned the same question to me, and he said he actually had a PhD student investigating this issue in the dissertation, but they didn't fully complete it. And then after that, I gave this question to one of my students to work on, two of my students to work on. But unfortunately, we didn't go much farther because I think my students were not really convinced of the value for the project. They felt like, okay, maybe the change will be small, even though conceptually makes more sense, much more sense to do a one step instead of two step. But if you're interested, we can discuss this further. Yeah, but I would say this two-step approach this way is not very standard. Yeah, it's not very standard, especially you have this outlier exclusion step, right? That will introduce more complexity. Like how do you define the thresholds? But basically, this is in the paper. Yeah. So I think the message I want to give you is actually I had this conversation from the editor I invited as speaker on Monday. She said after many conversations we had, she just felt like there are so many ways that statistics can go wrong. Basically, there's so many ways to do the analysis on the same data, right? And people may have very different judgments and the conclusions could change. Yeah, so we should be careful. At least if you learn this method from this class, that we should be careful when we do statistical analysis. I think that's valuable. Yeah. And also, even though we as statisticians, we may not agree with each other on how to do the analysis in the best way. So it's very driven by the question and also the data, right? The sample size is a very key factor because the sample size of your data determines how much information your data have. And the Bayesian approach, unlike the frequentist approach, is that the Bayesian wants to supplement the data with knowledge. You want to combine your knowledge with data. Then you can imagine the fewer data points you have, then the heavier role your knowledge will play. But whether you call it bias or not, that's another question, right, for scientists. So that's why I think it's all about how do we make decisions. And if you think about statistics as a way to do this, to make a decision out of data, then you won't be surprised that it's subjective. Because when we are shown the same data, maybe each of us will come up with a different conclusion, right? That's essentially what statistics is doing. But we hope to make it more principled. So that's why our guesses are not so, I would say, maybe sometimes your decision may be emotional, right? But here, at least we want to make it principled in somehow, yeah. All right, so this is from a work I authored with collaborators on this negative binomial assumption. I just try to draw your attention here again. I think I mentioned this briefly in the 229 last quarter. So about the negative binomial assumption, how critical it is, because if your data does not agree with this assumption, or this assumption doesn't seem to be reasonable on your data, then you will have some very surprising findings. And this work from published Genobiology is actually our surprise finding. And what we did was that my collaborators postdoc applied DE-seq to HR as the two popular methods to comparing RNA-seq samples from human individuals. And here, they are not replicates under two conditions. That's the biggest difference between this data set and the DE-seq2HR's original development. Because at that time, they were working on experimental replicates from labs, right? From under two conditions. So typically 3 versus 3. So they're replicates. And also, if you remember, last lecture, I talked about that measurement model and expression model, right? Together, you get observation model. So the measurement model, people assume that Poisson is reasonable, justified from the approximation to multinomial. So in that sense, you think, okay, I could use negative binomial as the observation model because I can assume the expression model is gamma, measurement model is Poisson. Together, I get negative binomial. But when your data are not experimental replicates about the same biological condition, because here you have patients, right? And patients are much more homogeneous. So therefore, for the expression model, let's say you know the true expression of every gene I in those patient samples, they are likely not gamma. If that's not gamma, then you wouldn't get negative binomial. So the distribution you get for counts can be much more widespread than the negative binomial. So the surprising finding we have is just from permutation. So we have 51 and 58 samples of two patient groups. It's about immunotherapy, the patients before immunotherapy and patients on immunotherapy. And the red dots indicate number of DE genes found by DE-seq2 and HR. All right. And what we play with the data is to permute the data. We just permute the samples between the two groups, but we still keep the two group sizes as 51 and 58. So basically, we just independently permute the, or I wouldn't say independent, we just permute the sample labels. So then we just randomly label 51 out of the total as group 1, the rest group 2. And then we run DE-seq2HR on the permuted data again. Then we find this distribution because we can permute for many, many times, and we did 1,000 times. And you can see that we get this bar as the average and this error bar as the standard deviation. So the surprising part is that on many permuted permuted samples, we can get even more DE genes. That's very surprising because we don't expect that, right? So that's in panel A. In panel B, we examine whether the chance that a gene is identified as DE gene in the permuted data is related to the rank. Okay, so sorry, let me see, number permuted, sorry. That's another data set, sorry. I should say the B first. So basically what we're showing here is that the y-axis is number of identified DE genes. The x-axis is the number of permuted data sets where a gene is wrongly identified as a DE gene. So you can see that for the DE-seq2 and HR result, some of their identified DE genes can be identified in many permuted data sets. The percentage here can be, we know this is like 40%, 80%, because we permuted 1,000 times. And this is just showing histogram distribution. For each, we just count how many genes are found in, how many identified DE genes by the method were found in what percentage of the permuted samples. So we see that this is quite surprising. And for some of the DHR genes could be even found in all the permuted samples. So it's not like, okay, they're only found from a very small percentage of permuted samples, but the percentage can be very high. And lemma view, which we didn't introduce, is another method. But I want to say that lemma was a method developed for microarray data for DE analysis, and it was based on just linear model, not so basically it's just when you say the generalized linear model, when you reduce this to linear model, you have the distribution as normal or Gaussian distribution instead of the Poisson or negative binomial. So it's a normal linear model for lemma, and Vium basically adapts this method to RNA-seq data. Okay, so we can see that it's actually relatively more robust, right, than the count regression. That's another count regression method. That's another very interesting observation, which is that even though our data may not be Gaussian-like data, for example, if you get a single-cell RNA-seq counts, you cannot assume them to be Gaussian because you only have non-negative integers. But some people's idea is to transform the data to be Gaussian-like. Then you could apply the methods developed by Gaussian for Gaussian data. And that's actually what the size factor normalization is doing. So you would actually be able to see that after you do this transformation, the the Zij here would look like more like a Gaussian for the cell across the cells for gene I, even though the original Xij are counts. Yeah, so probably I'll let you explore this. I'll let you try this on the first homework and see whether you actually see this pattern. All right, and furthermore, what we want to show here is that we want to look at the overlap of the DEGs identified from the original data and the permuted data. And we can see that basically the percentages for HR is quite high, right? So a quite large percentage of its identified DEGs were identified from the permuted data. Okay, in the D panel, what I want to show here is that if you want to use the log2 field change to filter out genes, if I say, okay, yeah, maybe that's true. Maybe they have too many false discoveries, but what if I add another filter using log field change? Unfortunately, on this data set, it's not that helpful because when we order the genes by log field change from the largest, the top rank 1 to the bottom, so we rank the genes, and we can see that the top fold change genes are more likely, actually quite likely to be identified from permuted data. So in this case, the log fold change is not an effective filter. And the real reason is because in this data set, given one gene, you may have some very large counts in some patients. So you will see very obvious outliers when you look at the DESeq2 or HR distribution. So outliers compared to negative binomial. So you have clear large outliers that cannot be explained by negative binomial, and they will have large fold change. And also when you permute them, these outliers will still be in one condition or the other. So they are still likely to be found. So that's the real problem. So biologically, I want to say why this actually matters or why is it dangerous. The reason is if you don't know this problem, if you don't know, okay, the D genes might be false positives, I look at the gene ontology enrichment analysis. I look at what GO terms, gene ontology terms are enriched in my DE genes. Then I can see that I get some interesting results. Like using these DESeq2, you have some immune response processes enriched. Using HR, you have some also terms related to immunity. Since the data set is about immunotherapy, you might be very happy to see the result and see, yes, there are indeed immune response differences between my treatment group and the pre-treatment group. But now if you know that these genes are actually, you can also find them from permuted data, then you might be worried, right? So I would just say that it's possible there are immune changes. It's very possible, but we just need to do a more careful analysis before we can conclude this. So the gene set enrichment analysis is not just enough. Yeah, but indeed, we check how well does the negative binomial fit to the data, and we do see a relationship. So that is for the genes that are identified from many permuted data, the fitting of the negative binomial is poorer compared to the genes that are rarely identified from permuted data. Okay. So you are still talking about this slide, right? So you are still asking about this. So when you say regularization, you mean this step? Yeah. So the question is, what is the cause of, yeah. But I want to say that when we do this, check this, we call this poor risk of fit because it's actually, we have a p-value for the goodness of fit test. And the smaller the p-value, the poorer the fit. So what we take here is the negative log 10 p-value. So that's why we call it the poor risk of fit. But back to your question, what is the cause of it? At least this shows the fit. This shows the fitted model from DESeq HR to the data. So we're not fitting negative binomial ourselves. We're using their fit and negative binomial. Yeah, so it's like an accumulative effect. Yeah, the model doesn't fit well to the data. Yeah. So what do you mean by true discovery set? Yeah, you don't depermute anything. Yes. Yes. So you're saying that we use the permuted genes as the use the D genes identified from a permuted as the background. And then we want to see how likely real discovery, like from the original data, original discovery is from the permuted set. Is that what you mean? Okay, but then your decision, if you think about, you mentioned hypergeometric test. If you use hypergeometric test, then you're not talking about individual genes, because a hypergeometric test is testing how likely is the set from this population. Right. So you're talking about these seq2 discovery set as a whole instead of individual genes. So you're answering a different question. The question you propose is whether seq2's discovery is not distinguishable from random discovery. That's the question you could answer using the approach you described. But it will not give me the information about how reliable each gene is. Yeah. Yeah, okay, you're talking about the pathway analysis. Okay, I see. So then you're saying that we could actually use the background as from the permuted gene. Yeah, I think that's a very good idea. Yeah, that's a very good idea. So you just use a different background, right? Yeah, thank you. Okay, so then let's continue. This is another thing I want to say about this paper, power analysis. So previously, when we do the permutation for all genes, then every gene becomes a true negative, right? Every gene is no longer true D genes. So when you have all genes as true negatives, yes, you can evaluate the false discovery rate, which is defined as the proportion among your discoveries that are false. But you cannot evaluate the power. Power is that among your true positives, what proportion would you discover on average? That's the power. But we don't have that information if the data only contains true negatives. So therefore, in this paper, we generated something we call semi-synthetic data, in which we have both true positives and true negatives. So how do we generate the semi-synthetic data? What we did is that on the real data, original data, we run the 5D methods, actually 6D methods at a very stringent FDR threshold. So like you can consider this to be a very small p-value threshold. We haven't talked about FDR formally yet. And we keep the discoveries for each method and we take the intersection. So we just assume that, okay, these are the genes that are likely D genes. We keep these genes data unchanged. And for the remaining genes, we permute each across the samples to make them true negatives. Okay, and then we run the methods and then we find this. So basically, the left column shows whether the actual FDR we evaluated with the ground truth as the true negatives we know, how is it compared to the claimed FDR threshold you want to achieve? And we found that there are inflations, but no inflation for Wilcoxon. And we found that if we look at the power, then Wilcoxon is actually among the top, because here the sample sizes are 51 and 58, very big. But I want to say that this is something very important pointed out by a reader, and they wrote a correspondence to the journal, which will appear soon with our response together. So they found that what we did in this analysis is unfair for the 5 methods except Wilcoxon. So Wilcoxon, we just run it directly, but the 5 methods like DESeq2HR, Each method includes a built-in normalization. And we apply the normalization as in the default pipeline to this data set and then find the genes. But the problem here is when we define the true positives and the true negatives, we are not assuming batch effects, right? So the true D genes are the original data, the other genes are permuted, but we don't assume batch effect. But you could assume that when you do normalization, for example, to make every sample have the same total, then you can make some of the non-D genes we assume as the true negatives become D genes. And then these genes will be discovered by the methods that did normalization. But in our ground truth definition, they were non-D genes. That's the problem. So basically, the readers point out that if we apply normalization to this semi-synthetic data set and then run Wilcoxon, then Wilcoxon will also have inflated FDR. So that's an issue pointed out by the readers. And this made us so I think it's a very important issue since we have been talking about normalization in this class. So now you can see normalization can really play a role in your evaluation. And so my take-home message, or in our response to the message, is that we would say normalization can cause false positives when true positives are defined without batch effects. So if that's how you define the true positives, then the data doesn't need normalization, and you shouldn't normalize the data. Otherwise, you will create false positives from the normalization. Yeah, and there's another paper I can discuss next time, which is something that has stimulated a very hot discussion online about a Nature paper about whether human microbiomes are the predictors of cancer. So that's some analysis. People have found, okay, the normalization also will make you have some classification algorithm that work very well. But basically, the separation of two classes were actually artificially enlarged by normalization. Yeah. So normalization is something very tricky. I want you to be careful. Yes. So which one? Not the paper and not the so you are talking about the microbiome thing, right? Yeah. Well, we can discuss this later. But I think my student has done some independent analysis on the same data. I can say that we haven't submitted our results yet, but I will say our results are more like in the middle. So we agree that the normalization plays some role there to enlarge to create false positives. But on the other hand, there's not completely no signal in the data. So there's still some signals in the data. It's just not as strong as in the original paper. So that's what we found. Yeah. But I think the rebuttal per se didn't address all the problems. Yeah, didn't answer all the questions. Yeah. But it's interesting. Yeah. Anyway, so I think there's something that we should be very careful about, especially for this past project. Well, we're still trying to make it a good design so we can really collect your honest analysis and feedback. And there's another interesting discussion on Twitter, like maybe 1 or 2 days ago. It's about this scRNA for R and scMPy for Python. Like they can give you very different results on the same data set. That's another issue. So really it seems that whether you use Python or R, you will make a difference. But that's not something we want scientifically, right? It doesn't really make sense that the pipeline can play such a big role. Yeah. All right. So hopefully I'll come back to the normalization issue later. Yeah. So then let's move on to this paper, bioarchive paper. And the interesting thing is that besides this phenomenon that, okay, using the SC transform Pearson residuals, you will still have this gradient in monocyte, right? This is what this group of authors pointed out. But besides that, there's a very interesting contradiction. So let's say this is the Surat, oh, sorry, this is the SC transform paper, the 2019 SC transform paper. So the argument is that when you do the SC transform, you can make the DE analysis more reliable because this is so-called the volcano plot for D analysis, where people look at negative log p-value in the y-axis, and they look at the mean difference in the x-axis. So sometimes people call this volcano plot because it looks like a volcano. And then you want to look for the discoveries that have large values in both, right? That means p-values are small and the mean difference fold change is also large. But this is what the authors wrote. This is how you would get from just doing log normalizations. You have so many D genes, but when you use the SC transform, you have so few D genes, right? The volcano plots look totally different. So the argument is that using SC transform, you can reduce false discoveries. However, from the same author's other paper, not the same author, same last author's paper 2 years later, which was pointed out by this group of authors, that the conclusion is the opposite. So here you find many more D genes after SC transform, but you found much fewer D genes when you just do the log normalization. So it seems that you have the opposite trend. I have to say, I didn't do the analysis myself, so I'm just quoting this bioRxiv, but it's so interesting. Yeah, I think probably I will let you try to reproduce this result in your first homework to see whether you can get the same result. And it's a way to explore the two types of normalization to see how they are different. Anyway, something I want to share from this bioRxiv preprint, and I have to say that this paper, even though it's posted in 2022, it's not published yet. So it's not really peer reviewed yet, but it's on bioRxiv. So the perspective is the variance stabilization. What is that? This is something I want to introduce to you as a statistical concept. Many people regard this to be a nice property and from Wikipedia. So the idea is that when you look at a random variable, for example, our gene count xij for gene I in cell J is considered as a random variable because there's measurement uncertainty, right? And we want to have the random variables variance independent of its mean. That's what we mean by variance stabilization. So you see that for Gaussian random variable, normal random variable, you do have this property. You have the mean, you have the variance, they're not related to each other, right? You can shift your mean, but the variance stays the same. But for Poisson, you don't have this because for Poisson, variance equals mean. So then it's not variance stabilizing because the mean enlarges, mean increases, variance increases. For negative binomial, you don't have this either, right? Because the variance has the formula as mean plus mean squared divided by dispersion. So it's depending on the mean. All right. So in the linear regression language, you may have this jargon again called heteroscedasticity. Very long word. What it means is that your error terms doesn't seem to have the same variance. Because when we look at a linear model, we typically assume, okay, the error term epsilon I are normal, mean 0, variance sigma square, right? The common variance is what we assume for simplicity, and that is called homoscedasticity. So you have the same variance for errors. But for Poisson, clearly, the larger the mean, the larger the variance. You don't have this. So hence, the variance stabilization transformation is that given we have the random variable x from Wikipedia has the mean mu and has variance depending on mu called h mu. Our goal is to find the transformation g of x and define this as y, so that for y, when we compute this variance, it's a constant. That's variance stabilizing. And this formula, variance of y is approximately h mu times the derivative, first derivative g prime mu squared. This is a result from an approximation called delta method, which I will not talk about today. But basically assume that we have this relationship. If y is gx, then variance of y will take this form, and h mu is the variance of x. Then if we want to find the corresponding g to make this a constant, it becomes an algebra problem. You just solve for the g, and this is what you get out of the g. And this was proposed in this 1936 Bartlett paper. So the goal, just to summarize, is that in our case, if we want to say the normalization is a good procedure from the variance stabilizing perspective. And this perspective can also kind of make your data like Gaussian. Gaussian-like because for Gaussian random variable, variance has nothing to do with the mean. Then we can apply techniques such as PCA to the Gaussian-like data, because that's what PCA is designed for. So anyway, so we will say that this is I think it's a useful perspective to think about, and we can use this to check whether, if we think about Y as our normalized data as a transformation of X by G, we can use this variance stabilization to check how good the normalization is. Okay, so basically going to check whether the variance of Y still depend on the mean of Y. Okay, then the question is, for the two normalization algorithms to talk about log1p CP10K and SC transform, do they achieve variance stabilization? And this bioRxiv paper explored this question. So I'm just taking the key paragraphs from it. The key thing is that, so something we didn't talk about called proportional fidelity. But the authors found that if you do the log1p, right, you do the PF, and then you plus 1, you take log, it's actually pretty close to the default of CRAD, log1p CP10K. So they are similar. And they found that SC transform doesn't give stable variance. So the sentence is, SC transform had the highest entropy of finding that can be explained by the heuristic clipping procedure performed on gene variances. That's the thing. So that's what we talk about for DC2. So the clipping of variances to the line, right? So, all right, but the finding is that this log1p PF method, which stabilizes variance after depth normalization, performs well in all metrics. So this is considered with another paper that considers normalization. So I feel like probably this kind of justifies why it's the default, because log1p PF is close to log1p CP10K, and log1p CP10K is easy to compute. So that's the default in Surat. Anyway, but still, the authors of the bioRxiv argue that indeed all current variance stabilization procedures are heuristics that ignore the fact that Poisson and negative binomial distributions may arise due to about physical stochasticity in bursty transcription and RNA degradation. So the transcription mechanism. The development of mechanistically justified normalization is a pressing challenge for single-cell RNA-seq data analysis. So yeah, basically there's no ground truth. Given that there's no ground truth, normalization, which one is bigger, which one's better, becomes an argument. And you can argue it from different perspectives, like variance stabilization. Okay, so then we will use the remaining time to quickly go through other methods listed on the 10x website. So I will only talk about one of the rest in detail, but I will just quickly glance through the rest. So these are all the ones developed in 2018 or earlier. So basics is a Bayesian method. And I think a key information I want to point out is that it tried to use some spiking information. So spiking is a word we often talk about in experiments and for computation analysis. So what I mean is that we try to add RNA molecules with known concentration to our sample. So then when we collect the data, we can compare our measurements to the known concentration as the ground truth or normalization. And this is a very standard procedure for bulk RNA-seq. But for single-cell RNA-seq, what is a reasonable or good enough spiking is still arguable. The reason is, for bulk RNA-seq, you basically try to scramble the cells, extract RNA, right, in one solution. You can add RNA molecules to the solution before you do sequencing. You can do that. But for single cell, the cells are in droplets. How can you essentially basically add spiking to the droplets that are like the RNAs in the cell? That's another thing. Because yes, you could add RNA molecules to the solution, and RNA molecules can be encapsulated into droplets. But that's not really equivalent to the RNAs in the cell, because the cell is still intact, right? Then you need to break the cell to get RNA. So there's the efficiency in that is not captured. So it's not that straightforward. Okay, SCnorm is a method I want to maybe spend a little more time on it because I think it's interesting technically. So here the idea is that SCnorm will try to use quantile regression to estimate the dependency between the log transform count and the sequencing depth. So still regression idea, like in SC transform. But I think the key thing is that it try to fit the model for each gene group. We will divide genes into groups, fit one model for each gene group instead of for all genes. I think that's the key thing. Okay, then what is quantile regression? As a very brief introduction. So the regression we often talk about is called the mean regression. So in the mean regression, what we did is that, so basically, when we talk about the mean regression in the linear model, we basically assume that the expectation of Y is some linear function of X. Okay, so I'll just quickly write it. Function of X. But when you see the quantile regression, you are replacing the left-hand side, not using the expectation, but by a quantile. So that's called a quantile regression. So you're still considering your Y as a random variable, but you're now assuming the mean of Y to be a linear function of some covariates. Instead, you're assuming, for example, the median of Y, the 75th quantile of Y, the 25th quantile of Y. So you're fitting a line for each quantile. So here is from Wikipedia. This one, the middle one is the median regression. So you're fitting the median as the left-hand side. And then you can have the quantile go down to 25th, 5th, and you can go up to 75th, 95th. So you're basically fitting, so given X, you're looking at the corresponding, this narrow slice of Y. So these are the values of Y, the distribution of Y given the X. So when we try to fit a line, it depends on what value we try to extract from this distribution of Y given X. We could fit the mean, we could fit the median, we could fit the quantiles. So then you may ask, what are the advantages of doing quantile regression rather than mean regression? I would say the short answer is similar to when we use median instead of mean. Okay, when do we use median? To be more robust, right? When there are outliers, we don't want the summary to be so sensitive to the outlier. But when you use mean, it's for sure sensitive. But when you use median, it's not so sensitive. Same argument here. So when you believe that your data contains some outliers, that you don't want to influence your regression line too much, you just use quantile regression. But nothing comes free. The price you pay is that quantile regression is slower. Yeah, the optimization is not as easy. Yeah, okay, so then let's look at the method. So let's pay attention to the notations again. So basically, SCnorm would require you to, let me erase this. SCnorm would require you to obtain log non-zero expression counts. So basically this YGJ, now we have a new set of notations, G for gene G, J for cell J. So you use this YGJ to denote the expression level of gene G, cell J after log transformation. So you can basically regard the data to be Gaussian-like. And this Gaussian transformation was very common in the old days for single statistical method development because it's easier. And you use XJ to denote the log sequencing depth of cell J. Previously, in the SC transform, this corresponds to log MJ, is that thing. And, all right, so K is the number of gene groups. All right, and this is the median regression. You have the quantile as 0.5, and you're assuming that the median of YGJ given cell J follows this linear function of cell J's sequencing depth after log transformation. Okay, so you see here the notation is right. There's nothing wrong with the notation. The model is specific to gene G, so you have G in the sub-index, and you also label cell J. All right, but then there's something I actually don't completely understand by reading. So basically, the author wrote, since the median might not best represent the full set of genes within the group. And since multiple genes allow for estimation of somewhat subtle effects, in this step, SCnorm considers multiple quantiles tau. So tau can be 75%, 25%, and multiple degrees D. So basically, you generalize your quantile to be something not specifically median, but can be any quantile, tau, and the case for group K, for the case group. And on the right-hand side, you generalize the linear function to a polynomial function. So you can have the degree bigger than dk bigger than 1. So it can be not just linear. But this is what I don't understand. The specific values of tau k and dk, the quantile degree, they call this chosen ones as tau k star, dk star, are those that minimize this thing, where this first term eta 1 hat tau k represents the count depth relationship among the predicted expression values as estimated by mean quantile regression using first degree polynomial. So this is from the first equation we saw here, is this one. They said that, okay, the slope for the depth, slope for the sequencing depth called tau, is called eta 1 tau k. But I don't get what you get from here. So the second term is mode G. And by mode, we typically mean the distribution, in the distribution where the peak is, right? That value is called the mode of beta hat G1. So I would assume that beta hat G1 is from here, but I'm not sure. So I don't know. And also, even that's my guess, I don't know why we can minimize this. And also, why can we get a degree, the polynomial degree here by minimizing this? So yeah, basically, I don't understand. I don't understand the rationale behind this choice of quantile and degree. If you understand it, you can let me know. Yeah, but anyway, let's just assume that they fit this model using this polynomial quantile regression. That's how they get a predicted YJ for YJ for cell J, gene I. And so what actually, I should add one more step in that. So they're not fitting this for particular genes, sorry. It's fitting across all genes for each cell J. So basically, I would say the model is across all genes in the same group. So that's why in this equation, there's no more index of gene G because it's for multiple genes in the same group. But once you get that, you can get the value for cell J, YJ, and then they basically try to derive a size factor in this way. So this denominator is from the observed data. You basically take the gene counts in the case group and you take the quantile of that. So for each cell, let me see, R is a tau quantile expression count in the case group. But this seems to be not related to cell J. It's across all the cells. But the numerator is for cell J, and it's using their predicted value from the quantile polynomial regression. And then they call this a size factor of cell J, but it's specific to the group K. Okay, and then if this gene G is in group K, then for cell J, you normalize it by dividing exponential of the level. So the reason they do this is because, remember, this YGJ is already on the log scale. So that's why they do it exponential to make it back to the count level, then divide by the size factor. So that's the whole thing. But one thing I can add about the notation here is that I think this SFJ should be SFKJ, because you'll get a different size factor for gene group K. And then when you normalize, it's depending on which group gene G is, then you divide it by the corresponding size factor. So having multiple groups is the key thing. All right, so before we wrap up, this is from the paper. So basically here, every dot here is one cell. And it's actually, I think they're putting genes together. So what they're trying to see here is that, okay, they do see the expression level have some relationship with the sequencing depth, but for the three groups of genes, the relationships are not the same. It has different slopes. So some gene groups have a bigger slope, some have a flatter slope. So that's why when they do the normalization, it's done by gene group. For each gene group, they try to fit a line so that after normalization, the log expression of genes in this group are no longer depending on the sequencing depth. It removes the sequencing depth effect. So you basically compare this to this. But this is from bulk data. For bulk data, they said the issue is not so obvious because you don't see that bigger differences among gene groups in the slope. The slope looks more consistent. All right, so that's SCnorm, normalized by gene group. And this one is SCRAM. And I will not go to the detail, but basically it's normalizing by cell pools. So I think the idea is that you want to pool the cells in some way and then normalize within the pool. So you have a pool-based size factor. And SCnorm has a gene group-based size factor. And this one, LINnorm, it's more complicated, has multiple steps, including filtering out genes. And basically, the highly variable gene selection is already here. But in the SIRAP pipeline, we haven't talked about highly variable gene selection yet. We are talking about normalization first before we define highly variable genes. Anyway, this one has the filtering of genes, and it has the normalization, which is basically considering the homoscedasticity. So the thing we talked about, and normality. Try to make data more normalized, and then do linear regression to remove the size effects. So sequencing depth. All right, finally, last one, SCnorm. This one uses a very different approach. It's trying to fit a power law Pareto type 1 distribution, and you will estimate the parameter alpha for this distribution using the maximum likelihood estimation, and then use this estimator as a normalization factor for multiplication. But I want to say that probably this one is purely based on this model. So I think the take-home message, several. One is that the log 1p CP10K is the SIRAP scan by default. It has some justification from the variance stabilization perspective. And there's no consensus on the best performing normalization method. Still open question. And it can be a good practice for users to try different methods and compare the results in the downstream analysis in cell clustering, embedding, which is like the 2D visualization, and differential gene expression analysis. And one thing I want you to be aware of is that each method has its own assumption, right? Relying on negative binomial, relying on Gaussian, or some Pareto distribution, and whether you want to group cells, filter out cells, whether you want to group genes, filter out genes. There are all these choices over there. Yeah, so that's just a summary. I hope now you see the complexity here. Anyway, so I'll see you on Wednesday. See you on next Monday. There is a chat in the box. Let me try to. Let me see. How do I get out of it? Thank you. Yes.